you're just joining us, we are going to wait a couple minutes. We'll let more join and we'll get started. All right, welcome everyone, and thank you for joining us today for Affinity Selection Mass Spectrometry from fundamentals to confident hit identification. Today, you'll get an overview of the ASMS workflow and a practical look at how Analytical Studios helps teams move from plate scale data to confident, decision-ready hit calls faster and more consistently. A couple of quick housekeeping notes. This webinar is being recorded and we'll send the link out to all registrants after the session. If you have questions, use the Q&A button at the bottom of your screen to submit them, and we will do our best to get to those at the end. Now, let me introduce our speaker. Don Nguyen is a senior scientist at Versidian. Originally from California, he did his undergraduate and graduate work there before heading to Germany for a postdoc, where he is based today. His background is in natural products chemistry and metabolomics, and a fun fact, he was actually a Versidian customer before making the move to the vendor side. So I believe this gives him a unique perspective when it comes to using these tools. I am excited to get into this topic, so I'm going to go ahead and hand it off to Don.
Great. Thank you very much, Ryan, for the introduction, and hello to everyone in the audience. I think actually I took a quick look at the registration list, and some of us have even spoken to one another in some capacity or another. So if I haven't met you, nice to meet everyone, and if I have met you before, good to see you guys again. Yeah, so as Ryan said, today we're going to be talking about Affinity Selection Mass Spec, kind of what it's used for, and more specifically about some of the improvements we've made to the software that help with ASMS analysis. Okay, so I just want to make sure that we are all on the same page. So let's go over a little bit of general background information for ASMS. Right, so ASMS is first and foremost a binding assay, and specifically we are looking for noncovalent binders. Right? ASMS can also be used in a low throughput mode or a high throughput mode, depending on how you have it set up for your particular system. The general workflow for ASMS goes like so. Right? So we'll take a target protein and then throw it into a mixture of compounds, right? This is something we call a multiplexed compound library. So we'll let this sample incubate for a specific amount of time, and then when it's ready, we will inject it onto a two-dimensional LCMS. The first dimension of LCMS is usually size exclusion, right? And what we'll do here is we will cut out our protein compound peak and then send it over to the second dimension, which is usually reverse phase chromatography. Right? This protein compound complex will dissociate because, again, we're under reverse phase conditions, so high temperature, low pH, aqueous organic mixtures. And then lastly, we have hit identification at the mass spec, right? So this is direct detection of our binders. So right, at the end of the day, ASMS is just another tool in the toolbox, so we should have some idea of how and when to use it, right? So some of the benefits of ASMS, this assay is run in solution and it's label free, right? All of this makes assay development very straightforward and very approachable. ASMS is also functional effect independent. So this could be a pro or a con, depending on how you look at it, fair enough. ASMS is binding site agnostic. And what this allows you to do is we can query multiple binding sites simultaneously. We can set up competition assays, for example, where if we have a known binder, we can block a binding site and see what compounds and at what concentration the known binder gets kicked out. And kind of along those same lines, we can also, at least potentially, identify unknown binding sites. ASMS is also very flexible. So depending on where you are in a campaign, right, you can deploy ASMS in various stages. So target tractability, primary and secondary screening, orthogonal confirmation, and then something we've, somewhat new for Versidian software is KD analysis, right? So again, ASMS is super flexible. As for some of the method's limitations, we will need sufficient affinity to detect binders at the mass spec, right? So I'm going to estimate here probably around double-digit micromolar affinities for some detection. And on top of that, binders will need to have moderate to slow off rates. So if you remember, there is some time delay between the moment you inject, go through the first dimension of LCMS, then the second dimension, and then finally detection at the mass spec. So if your off rates are faster than your injection times, right, we probably won't see that particular binder. ASMS is pretty protein heavy in terms of how much protein you consume for the assay, right? So certainly in a primary screening setup, you're going to be using a lot of protein. And then lastly, separation of our target and our binders from the non-binders is performed cold, so that's 4 degrees C usually, and performed in ammonium acetate, aqueous, and organic mixtures, meaning this assay isn't performed in buffer the entire time. These 2 points, right? These two have the potential effects of changing the shape and conformation and configuration of your protein, which has knock-on effects of your binding pockets on your protein. Right, so that is ASMS in a nutshell, right? Compound screening in mixtures. Your protein target or your biological target has to be kind of, let's call it size exclusionable, right? We need to be able to separate this complex away from small molecules and unbound compounds. And then lastly, we have hit identification in the mass spec. So one of the hardest parts with ASMS is certainly the data analysis, right? So in these various screening cascades, we almost always start with some sort of technical feasibility or assay development before we move into primary screening and then eventually hit confirmation. Usually at these early stages, and if you're lucky, you will have some tool compounds that you can use to validate your assay, right? But these are usually a low total number of compounds, so we don't need to do any compression. Now, compression is a term we use to essentially say, how many compounds are we mixing together in a single well, right? So at these early stages, it's usually just a single compound per well. However, once we go to primary screening, right, we're looking at a total of, tens of thousands of compounds, maybe even a full deck library, right? So that could be 1 million compounds or even a little bit more. So for an LCMS-based method to still be high throughput, we need to compress these libraries down into kind of smaller densities, right? Or larger densities, let's say. So routinely, we see anywhere between 100 and 500 compounds mixed together in a single well. And then at the extreme cases, we've seen upwards of 1,000 compounds into a single well. Once we get into hit confirmation stages, right, there's less total number of compounds that we need to analyze. So oftentimes we don't do any compression here, or you can do low levels of compression. Right, and then orthogonal validation, this is usually performed in other techniques. So functional assays, maybe structural biology assays, not anything that we need to think about for this particular discussion. Okay, so the challenges with ASMS are, of course, the data analysis. And this is something that Versidian has been working on for years now, right? How do we analyze data where you have potentially hundreds of compounds in a single well? How do we determine which of these hits are real or which of these hits are better than others? right? So already we have our standard ASMS capabilities, analysis capabilities, and our more recent KD analysis capabilities to address these two questions. But we're also trying to, always looking for better and faster and more accurate ways to do this. So the real challenge right now for Versidian is how do we automate our ASMS workflow, right? How do we make things smoother and easier to use and more approachable? So if there's anything to take away from this talk, it's this, right? We've been working super hard to streamline our ASMS analysis pipeline to get an easier to use product into our clients' hands and position these in such a way that our automation improvements can still be applied to screening data as well as more in-depth analyses like dose response data. Okay. To perform ASMS analysis, you firstly need Analytical Studio Pro. This is our core software. This will be usually sitting on a workstation, right? And we call this a standalone environment. So in a standalone environment, what you're going to need to do is acquire your data. This means creating your injection sequences, formatting your SI strings, and then actually acquiring the data itself. Just really quickly, SI strings or sample info strings are information that carry metadata about our sample. And then it tells Analytical Studio how to analyze that sample. So for example, one sample will have a plate barcode of 1, 2, 3, 5, 6, 7. This particular data is in a 96 well plate. There are 500 molecular formulas as part of the sample, right? So that's what these sample info strings do. They carry metadata so that AS Pro knows how to treat and handle that particular sample. Once data gets acquired, we're going to have to manually transfer this over to our AS Pro workstation. And then from here, with our compound list, we're going to create a targets file, load that targets file, and then open the data, right? So there's a fair number of steps that we have to deal with before we can even get to interpreting our data. And that's here at this final step, filtering and cross-site filtering. Now, we did have some attempts at automating this process in the past, but based on client feedback, right? It was honestly a little bit kludgy and kind of hard to use. And it was a halfway house of automation to the point where our clients preferred actually operating in a standalone environment rather than attempting our first pass at automation. But more recently, however, right, we've revisited ASMS workflow automation so that it is in better alignment with our other workflows like purification, like compound QC, right? So now this process is much more streamlined and much more approachable. So in order to automate ASMS, what we need to do is we need to change our environment, right? And this environment makes use of sample management services. So what SMS is going to do is it will consume a compound list and take all that information in the compound list and store it as Versidian metadata in its own database, right? The users then can access Simple List Builder to generate a sample list automatically, right? So we don't have to create those injection sequences manually anymore. These sample lists can then be imported onto our instrument along with the samples. And then as data begins acquiring, we're going to detect that data, copy it, and then transfer it over to the Express database. Right? Once data is here in Express, it'll undergo initial data processing. And then finally, a user can review the results and perform cross-site filtering, again, within AS Pro. So if we break down these steps, automated ASMS now looks like this. We're going to create a submission, generate our sample list, begin data acquisition, and then the initial analysis takes place automatically and behind the scenes. And then lastly, a user will have to perform cross-site filtering and generate their final results. right? So we're going to start looking at this in a little bit more detail now. So starting at the submission and sample list generation. Here's just, it's an example data set, and this is an example compound list, right? So really it's just a CSV, an Excel table with all the compounds we want to screen for, right? So here are all the various compounds, their associated molecular formulas, their locations on the plate. So in this case, right, all these compounds are being mixed together in well A3. A compound list can contain additional information. So here's a plate barcode, as well as a campaign number. You can also think of this as like an ELN number, for example. Now, what we're going to do is we're going to take the CSV, and then we're going to create a submission from it within ASPro, right? So we'll go to ASPro, create submission from CSV. We'll have a mapping file in place that consumes all of that CSV information and then puts it into the Versidian metadata format. So then what we need to do is just select our CSV. We can then preview our submission. So there's that ASMS campaign number, for example. There's our plate barcode. And as we drill further and further down into the submission, we can see all the samples we're going to be injecting, as well as all the compound information, molecular formula information, as it relates to that particular sample. Right, once this is ready, we can go ahead and submit this to create the submission, and then we will be generating our sample list from Sample List Builder. Right, so SMS again for CSV to submission, and then Sample List Builder for sample list generation. Right, within Sample List Builder, we're going to go ahead and select our submission, create our sample list, and really, you can just think of Sample List Builder as a wizard that guides you through a series of steps. So for example, if you are running multiple instruments in your lab, you'll have to select which instrument you intend to run this data on. And then we will have templates that need to be selected. These templates really just control the formatting of your injection sequences or your sample lists, right? So different vendors, they have different requirements for their injection sequences. We can pre-program all of that here in these templates. Right, so then as we step through the Sample List Builder wizard, we get a preview of our sample list. That will come up in just a moment. So here are all the sample names, their injection positions, injection volumes, as well as the instrument method that is going to be used. We can then save the sample list and then just import that onto the instrument PC itself, along with the samples. Okay, so now we begin the data acquisition and the initial analysis. So I don't have a mass spec in my living room, so I can't demo the data acquisition component. But again, once we detect data is being written to the instrument PC, we'll copy and transfer that over to Express. Initial data analysis and processing will take place behind the scenes. And what I can show you in the next few slides is interaction with the results, right? So from ASPro, what we're going to do is we need to search the Express database for all the data that we're interested in. From here, we can make a selection of which samples we actually want to view, and then we'll go ahead and download them into ASPro. Right, that'll take a few seconds, depending on how much data you're actually bringing in. But once data is actually in place, we can look at samples in multiple ways. So what I'm showing here is as we click on a particular well, we can see all the compounds associated with that particular sample. And then any of the samples, or any of the compounds, sorry, that are highlighted in green are the substances that we've actually detected in that data file, right? We also have standard LCMS functionality. So in a moment, we'll zoom in, zoom out to our chromatograms as well as our mass spectra so that you can investigate your data in detail. if that's necessary. But the whole point of our software, right, is that we can perform analysis in an automated fashion. So rather than look through well by well for every single sample, what we can do next is perform cross-hit filtering. And really, in a nutshell, cross-hit filtering is eliminating false positives and removing background noise so that any hits remaining in our hit list are high confidence. Okay, so here is our hit list before cross-hit filtering. And then once we click on this filter button right here, right, this recording was, it's being shown in real time, right? So just in a few seconds, cross-hit filtering is completed, which is much, much faster than our previous versions of ASMS, right? And to reiterate here, we have a fairly large number of compounds in our hit list. But previously, pre-filtering, we didn't know which of these hits were good and which ones were bad. After cross-hit filtering, which only took a few seconds, right? Our hit list now reflects just these strong hits shown in dark green, which I'm highlighting here. We can still interrogate the cross hits. So for example, here in sample position A12, this is our original hit, but what we can see is that this signal is also observed across almost the entire plate, right? And if we pull up these chromatograms side by side, their peak shapes, their peak intensities, peak areas, and retention times are all super similar to one another, right? This is what we call a cross hit. It's a hit that originates in one well, but is also observed in other wells. And since they look so similar to one another, we can't trust them. Right, so here's a closer look at all these filter settings. So first of all, to be considered a hit at all, you must pass these filters. So to be considered a hit, I have to have, my compound must be greater than 185,000 peak intensity. The compound must have better than 20 ppm error. And the compound, we should at least be able to detect 2 or more isotopes. Now, when it comes to the cross-hit filter settings down here, what these settings are basically asking are, compared to my original hit, how different are these other signals based on retention time? How different are these other hits compared to my original signal based on peak height differences, peak intensity differences, or peak area differences, right? So if all of these other signals show similar characteristics in retention time, in peak intensity, and peak area to my original hit, we're going to call this a cross hit. As opposed to this hit over here, originating in B13, these other signals have similar retention times, yes, but as you can see, their peak areas and peak intensities get smaller and smaller as you move across the plate, right? And since these signals are different compared to our original hit, we don't consider these cross hits. And if you really think about it, right, it's really just a little bit of carryover on the HPLC column. Okay, so that is, we just finished up the initial analysis and cross-hit filtering analysis within AS Pro, right? It's much faster than it was before. And then in combination with all these other automation improvements we've made, right? The ASMS workflow is just much easier and much easier to use, right? We can also apply all of these automation improvements on top of our KD analysis. So we still have to generate a compound list, create a submission within SMS, generate that sample list, and then acquire the data, right? So I'm not going to show these steps because really the new points are here within AS Pro and how our DRC analysis looks like, right? So again, we need to search our Express database for our KD data. We'll download those results into AS Pro. And we have a new panel now called our DRC results panel. So that's dose response curve results panel. Here over on the left are the what we call the KD results summary, right? So we have 4 separate DRCs here. As you click through everything, right, the visualization of the plate map and chromatogram and spectra get updated. Over here on the right half of this DRC panel are the individual items, the individual injections that make up this particular series, right? So these are all the varying concentrations of our injection. If we go ahead and display multiple chromatograms at the same time, right, we can even see a binding curve start to appear based purely on just a chromatographic visualization, right? But kind of the real component of our KD analysis is down here in the bottom with these plots, right? So we are plotting peak area and peak intensity information, as well as calculating the KD value, the dissociation constant value for this particular protein and compound. We calculate a Bmax value, as well as various statistics like R-squared values for your data. Right, so now this KD data can be used as a confirmation tool after hit identification, and we can use KD analysis to rank order which of these compounds is the best binder. Right, so in summary, most of our clients are still operating ASMS in a standalone environment, right? But we've made some really big improvements that include SMS and Express. When it comes to creating a compound list, right, this is still a manual step, right? We need to start this somewhere, so that's, manual compound list creation is unavoidable. But sample list and metadata generation is now automated, right? And this makes things so much easier than operating in a standalone environment. Initial data processing. When you open up your data in a standalone environment, right, that initial data processing takes place here within AS Pro. But now we can be a little bit more efficient with our time. So as the first injection is completed and the second one starts, that first injection is already making its way to Express and undergoing that initial data analysis, right? So we're being much more time efficient with this initial processing. Super big improvement here is within cross-hit filtering. So we're seeing 70 to 80% speed improvement for cross-hit filtering with the same results, at least in our test data set, right? So that's a really, really big leap forward. And then lastly is automation potential. Right? We've already baked in a little bit of automation when it comes to sample list and metadata generation, but now via SMS, which has API capabilities, we have the potential to integrate into electronic lab notebooks or IT systems or master schedulers, right? And orchestrators, right? So that really expands automation capabilities. And yeah, that's about it. Just want to wrap up here, right? So we've looked at ASMS, kind of how it's typically used in terms of identifying binders, how we can analyze dose response data within Versidian's Analytical Studio. We've made pretty big improvements to automation, right? So sample list generation, data transfers, and initial data processing. And then also some improvements to the core software itself, so cross-hit filtering and KD analysis. So again, thank you everyone for attending this webinar. Happy to take any questions if there are any.
Yeah, thanks, Don. Appreciate the presentation. It's definitely helpful to see it in action. So we can get into Q&A now. We do have a couple of questions already queued up that I'll kick us off with. But if you have any you'd like to ask, go ahead and drop those in the Q&A box at the bottom of your screen. So Don, first one for you. How do you define a recurring background signal and can I adjust it?
Right. So let me just jump back a few slides here because we can. Right. We control what's considered a background signal based off of our filtering settings. So yes, this is all user definable and user editable. It's a little bit hard to say to make like broad strokes because every sample is a little bit different. Every protein, every buffer is a little bit different. Instrumentation is also different. So I can't just say use these settings, it's good for absolutely everything. You need a little bit of fine tuning for your particular instrument and your lab. Um, but once you start seeing enough data, you'll get a good idea of what settings can be used here to define exactly for your situation what is a background signal or what is noise. Yeah.
Great. Thank you. Another one here regarding automated KD analysis. Can it be applied to rapid fire MS data?
Yeah, so I guess this is kind of a two-part question. We do have the capability, we say we're vendor agnostic, right? So we can look at Agilent data, we can look at Waters data and Thermo data. And then, of course, each of these vendors have separate instrumentation. We do support Agilent Rapid Fire already, so yes, we can technically read that in. We may need to finagle a little bit how the data gets split up and parsed out for KD analysis specifically. So that's more of a technical question and not a scenario that we've run into yet, but it is technically feasible in terms of bringing that data in and analyzing it. Yes.
Okay. Next one. Can this work on other ASMS approaches like with MALDI? MALDI.
Uh, okay, so this is actually an interesting question. So this is a paper that Boehringer Ingelheim had put out a few years back. They moved their ASMS activities outside of the conventional 2D LCMSs, right? And they plugged in their MALDI into an HTS line. Um, at the moment, we do not support any sort of MALDI data. We just haven't had a customer come to us and say that they needed Versidian Analytical Studio for their use with a MALDI instrument. So there's going to be a technical component of developing MALDI, reading the data out of a MALDI instrument. So that's one component. But assuming that technical hurdle can be overcome, in principle, mass spec data is just mass spec data, right? So yes, theoretically, it could be done.
Okay. Next question. Can I review removed cross hits?
Yes. So this is just a screenshot of the cross hits. Sorry, your hit list overall. These light green samples are cross hits, and then these dark green ones are hits. Users have the ability to manually toggle, right? We can manually override the decision that is made by our software. So if you find that one of these cross hits, or what our software has deemed to be a cross hit, you can always toggle that and flip the switch so that it's a genuine hit. And also vice versa. If there's a hit in here that should be a cross hit, you can toggle that as well. So yes, this is also user controllable.
Okay, great. And then we got one last one here. What vendor systems are currently supported?
hmm So I would say first and foremost, actually, you'll need for ASMS specifically, you'll want to do this on a high resolution mass spectrometer, right? So anything from a QTOF, a high res QTOF, or certainly Orbitrap's FTMSs. Um, so if we talk Orbis and FTMSs, we support Thermo. That's no problem. And then for QTOFs, certainly the major players, so Waters QTOFs, as well as Agilent QTOFs. And also we have support for Shimadzu instruments off the top of my head. Yeah,
Yeah, I'm sure there's quite a few, but all right. Well, that's great. We're going to wrap up the Q&A. Don, thank you again. Really valuable stuff, and we appreciate you
great.
taking the time. To everyone who joined us, thank you for being here. As a reminder, the recording will be sent out to all registrants, so keep an eye on your inbox. If you do think of a question after the fact, or you want to see Analytical Studio in action for your own workflow, there will be a link in the follow-up email where you can connect with our team directly. Thanks again, and I hope everyone has a great rest of your day.
Great. Thank you, everyone.